Um, Hi, everyone. Thanks for coming to today's seminar. Today we have Dr. Blair Canchero from Stanford University. Um, we are happy to have her to talk about decoding the music listening brain using electroencephalography. Um, Dr. Blair Canchero is an adjunct professor and Research Development Associate at Stanford University. Her neuroscience research investigates auditory and visual processing with an emphasis on multivariate EEG decoding and ecologically varied stimuli. Dr. Blair completed a PhD in computer-based music theory and acoustics at Stanford in 2016 and holds additional undergraduate and graduate degrees in music and electrical engineering from Stanford. She has held research roles both in academia and at music technology companies. As a woman in STEM, first-gen college student, and native Hawaiian, Dr. Blair is passionate about diversity and inclusion and is an active community organizer with the International Society for Music Information Retrieval, ISMIR, Women in Music Innovation Retrieval, WIMIR, at Stanford. Please welcome Dr. Blair. Thank you. I will give now the podium to Dr. Blair.
Thank you very much for the introduction. I want to just express how excited I am to be speaking with you today. I actually grew up in Eagle River, so I'm really happy to be with you all, even though it's virtually. So thank you for the invitation and for organizing this talk with me. So today I'll be talking about decoding the music listening brain with electroencephalography. So as this is an interdisciplinary talk, I'll just give a little bit of context about me and the research fields in which I work. So I'm currently at Stanford University. I am an adjunct professor in the music department with the Center for Computer Research in Music and Acoustics, or CARMA, and I primarily do music research as part of the Music Engagement Research Initiative at CARMA. I'm also a neuroscience researcher in the Stanford Graduate School of Education. So my first degree was actually in music. So I have a bachelor's in music. I worked for a little while, went back in music technology, and also did a master's in electrical engineering while I was doing my PhD. After that, I did a postdoc with the Center for the Study of Language and Information. And I've also worked as a research scientist in otolaryngology, head and neck surgery at the Stanford School of Medicine. And aside from academic work, I also worked at Shazam for several years while I was doing the PhD, and I worked briefly at another music technology company called Smule. And in terms of research areas, I work mainly at the intersection of two areas. So the first is music information retrieval, or MIR. So this is an interdisciplinary field that spans fields like computer science, electrical engineering, also musicology, and even library science. And one of the main focuses of this field is real-world application that can come out of understanding music and how it's used. So if you think about companies like Spotify or Pandora, or even Isotope or Steinberg, those are all considered core MIR companies. So my research in this field is really focused on the human aspect of MIR, and some things that I've been working on recently include study of collaborative playlists. So we look at how people interact with each other on streaming platforms and what types of social interactions take place. This was not intended to be about Spotify, but it turns out that it's largely about Spotify. So we're doing some studies in that area. When I worked at Shazam, I did some research on music discovery and trying to link discovery behaviors back to characteristics of the music at a large scale. I've also done some work on interfaces and custom interface design, mostly around perception and production of musical beat. And most recently, I've even gotten involved in some work with fandoms. So we have some collaborators outside of Stanford who are actually BTS scholars, and that's a really exciting new line of research for me. On the other hand, I also do neuroscience research, and I'm primarily focusing on multivariate decoding techniques. So what I'll be talking about today falls under that umbrella. I also do some other projects that involve machine learning with brain data, and that kind of feeds into a related topic of neural proximity spaces. So in these types of studies, we can take things like a classifier confusion matrix and convert that into a neural proximity space. And underlying my neuroscience research is some software development around analysis tools for brain data. And also, our group does a lot of open data. So we have a number of data sets of brain data that we release when our studies are ready to submit for publication. So my talk today will kind of be at the intersection of these two areas, and specifically we'll be looking at neural processing of real world music, so music that you would listen to in real life. So today I'll be talking about decoding the music listening brain. So let's first talk a bit about music. Music is a complex auditory stimulus, so there's a lot of stuff going on over time when we listen to music. And one of the ironies of working with real world music as my stimuli is that I cannot actually play them for you today because this talk is being streamed and it would flag the YouTube content filter. So my colleague Nick Gang has kindly given me an audio excerpt that we can use as an example throughout my talk. So let's have a listen to this. Okay, so presumably you were hearing that piece of music for the first time ever. However, you were probably able to grasp a lot of things about the music. So first, you might have noticed that it had a steady beat. You might have been able to make some judgments about its instrumentation, maybe that these were synthesized sounds and that the sound had a certain texture quality to it. You might have noticed how the different notes were changing over time, forming a harmonic progression. And you might have even, as I mentioned, made some judgments about the genre of the music and also whether you liked it or not. So this is a lot of stuff going on in a very short time. However, it's still not completely understood what our brains are doing when we listen to music. And this is especially the case for real world music, which is more complex and more difficult to map back to brain responses than the simple controlled stimuli. So the other aspect of talking about decoding the brain during music listening is to clarify what we mean by brain decoding. So for the purposes of today's talk, when I say brain decoding, I'm really asking, given a brain response, can we recover information about the music or maybe even information about the person who was listening to the music? So in this case, we're going from the response and trying to get back attributes of the stimuli or the listener. And this is a really interesting approach in my view because it really does enable you to ask a lot of different kinds of questions. So on the one hand, from a science perspective, we can use brain decoding to gain basic research insights into how the brain works and also how music works and how people interact with music. But on the other hand, from a more music information retrieval or engineering perspective, brain decoding approaches can also point to potential real-world applications. And finally, as this is an engineering talk, I'll be looking at approaches for analyzing brain data that are really treating our brain responses as signals. So for today's talk, I'll be presenting some recent and ongoing research that we're conducting at the Music Engagement Research Initiative at Stanford. And if you are interested in reading more about a broad introduction to this topic, we actually have a position paper that we published way back in 2015 that kind of lays out where neuroscience and music information retrieval can come together. So for today, I'll first briefly give an introduction to how we measure neural activity with electroencephalography, or EEG. From there, I'll go into research. So first we'll look at musical beat. After that, we'll look at how our neural responses can track time-varying audio features. Then we'll turn to the listener and look at how we can index listener engagement. And then I'll conclude with where we can go next with this type of research. So let's begin by talking about the measure itself, so electroencephalography. So what this is it's a measure of the electrical activity of the brain. And the way that it looks is that you would have something like this. So here we have a participant. The participant is wearing a stretchy net on his head. And what's happening is that each of these discs that are on the net has an electrode at the end of it. And the electrode is what is recording electrical activity that has propagated from the brain through the skull and the scalp. So this is just one type of imaging modality for brain data. It's the modality in which I work. And any brain imaging modality will have its advantages and disadvantages. So EEG is no different. In terms of advantages, it's non-invasive. So as you can see, we are merely recording from the surface of the scalp. So there's no need to go in under the skull onto the surface of the brain here. EEG is also known for having superior temporal resolution. So what this means is that we can record brain activity on the scale of milliseconds, which is especially useful when we think about something like music, because musical events are also unfolding on the scale of milliseconds. So it's well matched in that respect. Compared to some other modalities such as functional magnetic resonance imaging, EEG is relatively inexpensive and it's also fairly portable. And finally, as you can see, we're able to collect data from sensors that are covering a good portion of the head. And so we consider this to be a wide field of view and whole brain coverage. On the other hand, there will also be some disadvantages. So the spatial resolution of EEG is not so good. So if your main interest is in knowing where in the brain processing is taking place, this might not be your go-to modality, although it can be done. It's important to note also that EEG data are very noisy. So we're operating at a pretty low SNR of around 20 dB. And so what this means is that your signal of interest will often be about 1/10 of the amplitude of the surrounding noise. And finally, we will have a high data dimensionality. So in this case, if you record at a kilohertz, and if you have this sensor net, which has 128 electrodes, you're getting over 100,000 data samples every second. We can look a bit at the characteristics of the data. So every sensor will record a time series of data, and we're in a fairly small amplitude range of microvolts. And this is an example of a readout. So what's happening here is every row is a time series recorded from a single electrode. Most of the cortical activity that we're interested in is in the frequency range below 50 hertz. And it's important to note that when we experience a stimulus, our brain response is not instantaneous. So there will be what we call a stimulus to response latency between the stimulus event and the corresponding brain response. And for cortical responses, that'll typically start around 80 milliseconds and can continue into several hundreds of milliseconds after the event. And it's especially important to note that particularly for these complex real-world stimuli, we probably won't know the exact latency in advance. And we'll actually come back to this point later, so I'm just going to put like a little star. So just remember, we don't know our stimulus to response latency. But in general, I think a useful way to think about EEG is as a collection of signals. And as you can see, there's some oscillatory activity going on in our image here. And therefore, these signals can be analyzed in the time or the frequency domain. So I'll be focusing mostly on research findings today, but I just wanted to point out that underlying our analyses are a lot of matrix factorizations. So as I mentioned before, we do have these challenges of working with EEG. So we know they're noisy, we have a high data dimensionality, and as you can imagine, electrodes that are close to one another are probably recording activity that's fairly correlated. So one way that we address this is by factorizing our EEG matrices, and specifically we're using eigendecomposition techniques to do this. And so what we're going to do is we're going to reduce our dimensionality across the space dimension, so across the electrode dimension. And what we'll be doing is computing optimized weightings to apply to our electrodes so that we can take a weighted sum. And these optimized linear weightings are referred to as spatial filters. So if you're used to working with matrix factorizations, it might be more useful to look at it this way. So now we have every electrode of data is represented in a column of this matrix, and we're essentially going to apply matrix factorizations to compute weighted sums of columns. And that will reduce our dimensionality from many electrodes to one or just a few spatial components. And as these are eigendecomposition techniques, these approaches will produce multiple solutions. But for today, I'll just be talking about vector representations of the first components. So if we're doing principal components analysis, I would be looking at just the activation in the space of PC1. So that was just a very brief introduction to EEG, and now we'll jump into some musical topics. So let's first do a little listening exercise. I would like you to see whether you can tap to the steady beat of the music. All right, so if you could tap to the steady beat of the music, you were probably tapping at a rate of 120 beats per minute, which is the tempo of this excerpt. And if we do some dimensional analysis, we would find that that translates to 2 hertz, so 2 taps per second. And since this is electronically produced music, we would say that our stimulus has a periodicity of 2 hertz. So this is actually a useful thing for us to work with because stimulus periodicities are of special interest with EEG data. And the reason is that if you have a stimulus that is periodic, you will see that same periodicity in the EEG response. So this is called the steady state paradigm, where you can identify stimulus periodicities fairly easily in the EEG spectrum. So this is just an example from a recent review paper by Anthony Norcia and colleagues. So what they're showing here is an EEG response to a visual stimulus that was presented at 7.2 hertz. So that means that there was an image or some kind of visual input that was flashing on screen 7.2 times per second. And the figures are showing the EEG in the time and the frequency domain. So on the left, we have the time domain. So we have time in seconds on the x-axis and then our microvolt amplitude on the y-axis. And you can see that there are clearly oscillatory components to this response, and that there are, I'm guessing, 7.2 prominent peaks occurring in the space of 1 second. So that's time domain. We can also take the FFT and look at the magnitude spectrum in the frequency domain. So that's what's shown on the right side. So now we have frequency on the x-axis and then Fourier amplitude on the y-axis. And what you can see is that there are these very strong spectral peaks coming out of the noise floor at 7.2 hertz, as well as integer multiples of that frequency. So the steady state response is most often used for vision research, less so in auditory research. However, as we noted earlier, music can have stimulus periodicities as well. And there is a body of research that has shown that when you listen to simple synthesized rhythms, you will see spectral peaks in the EEG at the beat frequency. However, this approach has not been used so much in response to real-world music, where, as you can imagine, the spectrum might be more complex, and therefore we might not expect to see such large peaks in the EEG response. And in terms of real-world application, this is kind of an interesting question because tempo detection from audio is actually an open topic in MIR research. So while we as humans can easily identify a beat and tap along to it, it's still a challenge to do that from audio. So findings in this area could inform that. We could also even think of something like a brain-based tempo detection system. So our first effort in this area came actually out of a dataset paper that was led by Stephen Losarelli and published in 2017. And for this first attempt, we were just trying to characterize the data. So we just did a simple FFT approach. So we began by looking at the stimulus FFT. So what we have here are 3 songs, and each song has a different tempo. And we extracted the amplitude envelope of each song and then just plotted its FFT. And the beat frequency is the dashed green line, and then multiples of that are the dashed lines at higher frequencies. So we can see that each song has a number of spectral peaks, and those spectral peaks can fall on the beat frequencies, but there are also other peaks that are at other frequencies. So we recorded EEG to people listening to these songs, and we looked at the FFT of the response. So our first attempt was not exactly a spatial filter because we weren't optimizing anything. So we just took the mean of the data matrix across all of the electrodes. So that produced a vector, and then we just plot the FFT of that vector. So we have frequency here on the x-axis and Fourier magnitude on the y-axis, and it's not actually looking very good. We see some evidence possibly of peaks at the beat-related frequencies, but as you can also see, there's a pretty substantial noise floor, and this is just a known characteristic of EEG that you'll have a lot of noise, especially in this low frequency region. So maybe averaging wasn't the best thing to do. We tried something else that was a little bit more sophisticated. This time we do PCA, and we plot the FFT of the first principal component. And in this case, we can actually see some peaks coming out. So what we see are, they're more robust, so they're coming out of the noise floor. And moreover, the peaks are really locking in to the frequencies that are related to the beat. So this is kind of an important point because it's showing that while the stimulus spectra had peaks at the beat and elsewhere, it seems that our brain responses are really locking into just those frequencies that are related to the beat. So this was an encouraging finding, and we followed up on it in a slightly different way in our most recent paper from this year. So now we take a coherence approach, and we're specifically computing coherence between the EEG and the stimulus envelope. So whereas with FFT, we were just looking at what frequencies were present, now we can actually ask which frequencies in the response relate to the frequencies in the stimulus in a meaningful fashion. So here's another, this is a different song with a different tempo. And in this case, we computed magnitude envelope fluctuations and then plotted the FFT. For this study, we used a different spatial filter, so not PCA, but something similar where we are actually filtering to maximize correlation in time across different trials of EEG. So we're not filtering for beat, but this approach would still help us reduce our data dimensionality and pull out some meaningful signal that's related to the stimulus. And what we did is we took our EEG trials and we computed the magnitude squared coherence between every trial and our stimulus envelope. And then we also computed the cross-power spectral density phase angle between each trial and the envelope. And this would give us a sense of the lag between envelope events and the corresponding EEG response. So here's our result. So in these plots, we again have frequency on the x-axis, and in the middle plot, we have stimulus response coherence on the y-axis. So you can think of that as like a measure of correlation between stimulus and response at each frequency. And then in the bottom plot, we have those CPSD phase angles. So these are on a radian scale. So you can think of this as kind of like a lag between stimulus and response. So a couple interesting things come out here. So first we see that we have a really large coherence peak, and importantly, that peak is locking on to one of the harmonics of our beat. So in this case, it's at twice the beat, so maybe twice the rate at which you would tap to the beat. And we note that this is not actually locking onto the beat frequency. As we discuss in the paper, we think this might relate back to our spatial filtering technique. Another thing to note here is that whereas on the previous slide we had that large noise floor, the coherence approach actually just flattens out the noise floor. So what this is saying is that while we had a lot of low frequency activity in the EEG, it was not activity that was related to the frequency content of our stimulus. And finally, we can see that the distribution of our phase angles, it becomes very low variance at the frequency of the high coherence. So what this is saying is that, it's basically just enforcing more that when we listen to music with a steady beat, our brain responses are not only oscillating at the frequency of the beat, but it's also phase locked to the beat. And that phase is consistent across different EEG trials. So this is an ongoing research topic for us. We have a couple of projects going on. So we think that we've established that we can find these spectral peaks related to beat even in response to real-world stimuli. But as I noted, we're not actually trying to spatially filter the data yet to optimize on that feature. So we were either trying to maximize proportion of variance explained or maximize temporal correlation, but not really the steady state activity. So one thing we're doing right now is we're working on a new spatial filter, and this one will actually be computing the optimal electrode weights to maximize neural activity at a known frequency of interest. And we're getting some promising results in this area so far. And then in a separate project led by Jay Apogee, we're looking at beat in a slightly different way. So not at the frequency domain representation, but actually in terms of computational models of beat perception that are derived from the audio. And we are comparing the output of these models to different characteristics of the brain response. So that wraps up our beat section. Next, we'll talk about tracking of audio features. So when we listen to music, we're listening to acoustical events that unfold in time. And when we record brain responses with EEG, we're also these are also brain responses that are unfolding in time on a similar time scale. So we might want to know whether we can actually correlate these two measures. So when I talk about time varying audio features, I'm referring to any time varying feature that you can extract computationally from audio. So envelope is one example. You can think of something like spectral flux that's computed in short overlapping time windows. And in fact, there is a whole variety of audio features that have been developed specifically for the analysis of music. And what we want to do is look at how those features correlate with the EEG response. So if you think of something like audio envelope over the course of a song and the response you record from a listener who hears that song, we're just trying to find a way to optimally map these two types of data. So up to this point, I've been talking about spatial filtering. We will also do a spatial filter here, but we're going to do a second filter along with our spatial filter, and we're going to use canonical correlation analysis. So the specific approach that we use for this was introduced in a 2018 paper by Jacek Machovsky and colleagues. This is an implementation specifically for EEG. So what you have here is you'll have your EEG data matrix. So we're in the column form where each sensor is a column. And then we'll also have our stimulus feature vector. So this vector could be something like the audio envelope that I showed in the previous slide. And importantly, both of these data variables are the same length in time because we want to correlate them in time. You may remember from before with my starred point that there's a latency between stimulus events and the corresponding brain response. And we do not know in advance what that latency is. So we can actually write this into our computation. So what we're going to do is we're going to take our feature vector and we're actually going to expand it into a feature matrix. So what's happening here is that we're making incrementally delayed versions of our feature vector in each column. So column 2 is the feature vector that's delayed by 1 time sample, and then column 3 is delayed by 2 time samples. And by doing this, we can actually impose all possible delays up to, say, 1 second or 2 seconds. And what the canonical correlation analysis procedure will do is it will first do our spatial filter. So it'll find the optimal weighting of columns of EEG. But it's also going to find the optimal weightings of columns of this feature matrix. So what this means is that it's going to find the optimal delay between our stimulus feature and the corresponding brain response. So this delay weighting you can actually think of as an impulse response on your stimulus feature. So we tried this out in a 2017 paper led by Nick Gang. So we did a variety of different features. The figures I'll show, I think, are for RMS that was computed from the audio. And we did find significant stimulus response correlations during music listening. So we do find evidence that the brain is tracking these musical features in time. And one interesting finding that came out of this is that we can actually look at our filters. So if you think of a spatial filter, it's a vector. The length of the vector is your number of electrodes, and each element is a positive or negative weight. So we can actually visualize those vectors on a map of the head. So that's what's happening here. We have the nose at the top, the back of the head at the back on the bottom, and then the ears on the side. And it's showing us where we were either going to apply positive weights in red or yellow or negative weights in blue to reduce our data matrix from electrodes to components. So we had 4 different songs, and we computed CCA separately for each song. So the first thing we see is that the spatial filters are actually very consistent up to a sign flip, which is fine. And this makes sense to us because that would imply that you're using this, the same brain region to listen to music, no matter which specific piece of music you're listening to. We can also visualize our impulse response. And in this case, we took our stimulus filter impulse response and visualized it in the frequency domain. And this was another interesting finding in that while our spatial filters were consistent across songs, our optimal delays were song dependent. And on top of that, the frequency activity in these impulse responses was peaking at frequencies that related to the musical beat. So this means that when we listen to music, it's not that there's always the same amount of optimal delay between the music and our brain response, but that it's actually implicating the beat again. And so this provides further evidence that when we do listen to music with a steady beat, our neural activations are really locking on to those beat-related frequencies. So there are some interesting applications you can do with this approach. One is automated music annotation. So if you get to a point where you can basically recover musical features from the brain response, you could use that to label music. In addition, this approach is very interesting because you are correlating the brain response directly with the stimulus. And while we have used the approach just to look at things on a group level, so like what is our group of listeners doing, you can actually use it to study brain responses to unique perceptual experiences. So in the 2018 paper that introduced the CCA EEG method that we use, the researchers actually collected EEG while the participants were playing a video game. So in this case, you can imagine that everyone would be having a different experience because they're controlling the game. And they were able to get significant stimulus response correlations. On top of that, the extent of the correlation is known to reflect attentional state. So there would be any number of applications that could build off of automated detection of a person's attentional state. So that was tracking of stimulus features, and we'll now move on to our final topic, which is about indexing engagement. So everything I've talked about so far has been focused on connecting the EEG response to some characteristic of the stimulus. But there are other factors in this picture, particularly the person who is listening to the music. And so we might want to ask questions about the listener's state and specifically whether the listener is in a state of engagement. So the applications in this area really go into the user direction. So if you think of anything about modeling a user or a listener and their preferences. So this could feed into a recommendation system. If you are a music creator, you might want these insights to aid your compositional process, to engage the listener in a specific way. And these types of findings could also inform what we call music for a purpose. So if you're using music to regulate your mood or to increase your productivity. So the neuroscience of engagement is actually like its own active area of research. So to kind of get us situated in this way of thinking, let's first define what we mean by engagement. So as you can imagine, there are many ways to engage with music, but we're specifically interested in the state of focused engagement. So we found this quote from Emery Schubert and colleagues to be very helpful in framing the concept. So we think of engagement as being compelled, drawn in, connected to what is happening, and interested in what will happen next. And so we formulate our research questions around the following line of thinking. So when someone's in a state of focused engagement, their brain response, which is partly driven by a stimulus, partly driven by other things, will become more driven by the stimulus than usual because they are so focused on that stimulus. As a result, if you have an audience who is all focused on the same stimulus, that greater stimulus-driven component in each of their responses will mean that their responses are more similar to one another than they would have been if they were not in a state of focused engagement. And consequently, if you were to correlate the responses, the correlation would be higher. So the correlation is actually what we use as the metric of engagement. And specifically, this is referred to as the intersubject correlation, or ISC. So if you think about this practically, if you were conducting an experiment, you would have an audience of some kind and a stimulus that they would all experience, and you would collect their response measures to the stimulus. So you could do this, everyone at the same time. Typically with brain recording platforms, you can only do one person at a time. So we would do this asynchronously. But the important thing is that everybody experienced the same stimulus. After that, we have our spatial filter. So we're going to compute our electrode weightings to maximize correlation in time across the different trials of EEG to the same stimulus. So in essence, we are computing a spatial filter to maximize the ISC. And then once we have projected our data from electrode space down into the vector space of the component, we'll correlate those response vectors. And then the higher the correlation, you would interpret that as the higher the state of engagement. This approach is really interesting to me because it's just so data-driven. So one challenge of working with complex real-world stimuli is that you can't always know in advance what the response will look like. But to me, this method is very powerful because you actually don't need to know anything in advance of what you think the brain response will look like. You're just looking for brain responses that are the same or similar across your different trials of response. So this is not a new line of research. It's been going on for almost a decade now with EEG. And there are a number of studies that have verified that the ISC can index states of engagement or outcomes related to engagement. So ISC is known to be higher when someone is paying attention to a stimulus versus distracted. It's known to correlate with learning outcomes. So if you watch a video and take a quiz, it will predict your test score. And ISC can also predict longer-term memory retention. In a study on viewing of film excerpts, it's been shown that the intersubject correlation will be especially high during periods of high suspense and tension in a movie excerpt. And finally, there's some evidence that the ISC of a small sample can actually predict the preferences of a whole population. So in this 2014 study, the researchers found that the ISC of fewer than 20 people could predict Nielsen television viewership over the course of a TV episode and could also predict Facebook ratings of Super Bowl commercials. So when we learned about this approach, we were very excited about it, but we also weren't sure if it would work with music. So considering the studies I just mentioned, those were all about stimuli that were basically trying to convey a pretty clear narrative or message. But music isn't always like that. So most music is actually not communicating an explicit narrative. It's more abstract. And as you may know, like if you reflect on your own music listening behaviors or maybe the preferences of your friends, individual listeners can engage quite differently with a given piece of music. But even so, we thought that music would still provide some frameworks in which people can develop expectations. So if you think about Western tonal music, so this could be like classical music and a lot of pop music, the musical events are structured in time and also in their harmonic progressions and melodic activity. And the way that these things are organized in time lets the listener do things like build expectations, have those expectations realized, or even experience surprise. So we thought that those types of devices might be analogous to the narrative devices that have been shown to elicit high ISC in previous studies. So this is our first major publication around ISC. It just came out earlier this year. And we had a few questions that we were trying to address in the study. So the first was, would there be significant ISC at all during music listening? Or is this just for movies and speeches and not for music? Our second question had to do with time. So we are very interested in the way that music is not just random sounds, but sounds that are intentionally structured in time to form a musical work. And so we wanted to know whether if we disrupted that temporal organization, if it would impact this neural metric of engagement. And finally, we wanted to know whether engagement would align with enjoyment, because those seem like similar things, but maybe they're not the same thing. So for this study, we had 16 stimuli. So we had 4 songs in 4 conditions. These are the waveforms of one of our actual stimuli. So the first condition is intact. This is a complete full-length song in its entirety. We also have reversed, so the waveform is reversed. And then we have a measure condition where we cut it up into groups of 4 beats and shuffled it. And then our final condition is phase scrambling. So this is our most extreme condition. And what we did here is we took the FFT of the audio, we randomized the phase at each frequency, and then we converted back to the time domain. And again, I can't play my actual stimuli, but we can listen to some manipulations of our example piece. So here's the intact. I think you know how the rest of that one goes. Here's the reversed version of that. Okay. In this version, you'll hear the musical content kind of jumping around every 4 beats. This is the measure. And finally, here is the phase spectrum that's been randomized. All right, so those are our 4 conditions, and we actually used Bollywood songs for our stimuli. You might wonder why we did that, and we actually did have good reasons. So we wanted songs that would be new to our participants, but had been proven to successfully engage a large audience, first of all. Secondly, these would be broadly in the pop genre, so we felt that a listener could kind of grasp what was going on in a first listen. And importantly, our participants could not speak Hindi, so this enabled us to manipulate the stimuli in time and not have people be able to understand the lyrics in one condition, but not another. And all of these songs were 4.5 minutes long. So whatever you were assigned, you listened to it for 4.5 minutes, even if it was the phase scrambled one. So we collected data from 48 participants who self-reported that they liked various genres of music. And we had 4 songs that these were based off of. So everybody got 1 song in a different condition and heard each one twice. And then they also delivered ratings about pleasantness, musicality, order, and interestingness, which we took as kind of a proxy for enjoyment. And what we expected to see is that the intact versions of the songs would elicit both the highest neural metric of engagement and the highest enjoyment, since these were the versions that were intentionally composed for consumption. So first we can look at our behavioral ratings. So here we have each of our 4 questions, and every bar is a stimulus, and it's colored by stimulus condition. And indeed, our participants preferred the intact versions of the song the most. And our true control with the phase scrambling received lowest ratings, and then the measure and reverse conditions were kind of in the middle. So this was pretty aligned with what we thought would happen. And so we were encouraged by this result and thought that this would also come out in the brain data. But in fact, this was not the case in the brain data. So here we have the intersubject correlation for each stimulus, and any bar that comes above the shaded gray area represents a statistically significant result. So it was not the intact version, but rather the measure version that was the shuffled at the unit of 4 beats that had the highest ISC. The phase had the lowest ISC, as we expected, and then intact and reversed were in the middle. Reversed was slightly higher than intact in this case. So this was not what we thought would happen, but it actually reveals some interesting points about music listening and the temporal hierarchical structure of music. So our first conclusion is that engagement is not the same as enjoyment. So that was a helpful thing to verify in this context. And then when we look at the highest ISC for the measure condition, we think that it's really hinging on the way that musical events are structured in time. So when you think of a song, the song can be broken down in time into sections, and then inside of each section you might have phrases, and then phrases comprise measures, and then measures are made up of beats. So that's a very simplified temporal hierarchy of music. But when we imposed the measure condition, we were essentially wiping out these higher level structural elements of the song. And as a result, the entire temporal framework was hinged on the measure level, or these groupings of 4 beats. And we think that due to this, the listeners were able to come to expect new musical content every 4 beats, because it would always skip around in the song. And we think that it's actually this aspect of not only knowing that new content is coming, but knowing exactly when it's coming. So we call this a strongly timed expectation for new content. And we think that it was the fact that these were strongly timed that kept listeners in an engaged state while they were listening to that version of the song. So this opens a lot of really interesting questions that we can explore further about music and time and structure, and also novel events in music. So that was our first project. We have some other projects going on in this area. So we have one study where we can look at ISC over time and see whether the engagement metric becomes especially high as you approach very dramatic high points in a musical work. For this study, we also have physiological responses and continuous behavioral responses. So we're excited to do ISC analyses on those. In a separate study led by Tyson Dower, we're looking at a completely different genre of music. So this is a genre called minimalism, and our main stimulus here is a 5-minute excerpt by from a work for piano, which is essentially this 12-note motif that you see on the right that's just repeated over and over again for 5 minutes with very subtle temporal shifts. And so we're interested to see whether the ISC can actually capture engagement with something that is so extremely different from the kind of Western tonal music that we've been looking at so far. So this brings me to the end of my talk. So I hope if you could take anything away from this, it's first that music contains complex acoustical events that unfold in time, and it's really a remarkable feat that our brains are able to process music. And I hope that I've also shown that by adopting these signal-based approaches to analyzing EEG data, it can actually help us conduct research in this challenging scenario where we're working with complex real-world music. And so our group is really excited to be kind of taking the methodologies that we've been building up over the past 5 years to look at new questions. We're also looking into new methodologies. So how can we represent music better? Are there nonlinear ways of approaching the data, for instance? And I was first drawn to this line of research because I'm very interested in studying how we experience music in real life. So I'm always interested in things that point toward more ecological validity. And I just wanted to point out that mobile EEG has actually made incredible advances over the past decade. So for about a tenth of the price of a research grade setup of the kind we've been using, you can actually collect data with fewer sensors out in the world. This example headset actually has like a VR goggle interface. And so I think this is a really exciting direction that this research can go to study music consumption in the real world. So that's the end of my talk. Thank you for listening to me talk. This was a very, very collaborative set of research projects that have been going on. So I just want to thank the people who I've been collaborating with on these studies, whether as students or colleagues or mentors, and also want to acknowledge my funding sources. And that's the end. I'm happy to answer questions. We have a lot of public data, so I'm also happy to discuss collaborations or other research directions. Thank you.
All right, thank you so much. So we do have a few questions to get kicked off here. One of the questions is a faster or slower beat better for gauging brain response?
Oh, that's a really interesting question. So with the steady state response, so two things. With steady state, there's kind of a preferred range of frequencies that come up a lot in research. So maybe from like 4 to 7 hertz or so is often where you'll get especially robust brain signals. So if you can get a beat that lies in that range, then you might be in good shape. There are also perceptual limits to how we as humans perceive beat. So if the beat is extremely slow or extremely fast, we actually won't entrain to it the same way that we do with beats that are in a more moderate range. I'm sorry, the exact speeds escape me right now, but I can look into that.
No worries. No, 4 to 7 hertz sounds like a pretty good answer to me. Are there particular types of music that are better for gauging brain response? I'm guessing maybe genre.
Oh, that's a good question. The use of real world music in neuroscience research is so small at this time that it it seems like we're just kind of getting started in this area. So, like we've used a few different genres. There's another group that uses a tango piece a lot. There was a recent study with ISC that used kind of a variety of genres, but I think we still have a long way to go. And I think it's also an interesting direction of research to look at people's specific preferences for the genre. So there's some research that shows that fans of heavy metal perceive emotion in that music differently from non-fans. So I think that asking a question like that with the brain response could be a really interesting area for research.
The next question is reads as follows: is there any case in your music listening work where EEG ISC can't be established at all?
Not yet. We tried to start out with music that was fairly accessible to our listener pool. I'm interested to know whether, if I go back to this piano phase study, since that type of music is so different from the conventional Western music, it's kind of an open question at this time whether we will see the ISC for that particular piece.
All right, and I think we have one more question. It is theorized that musicians have higher neuroplasticity than non-musicians. Is there any evidence to your knowledge of higher neuroplasticity among those who listen or dance to music of any variety?
Yes, there's actually, that's a question that kind of comes up in a lot of different facets of music neuroscience research. So there's evidence of it. So I work in cortex, so there's evidence in cortex, but there's also evidence, even if you look before the cortex, to really low-level auditory processing, like our auditory brainstem. There's some evidence that trained musicians, among other populations, will actually have more robust processing even before the signal reaches the cortex.
Is that true of non-musicians?
So I think the findings are that musicians compared to non-musicians would have maybe a more robust response, but also people who speak tone languages like Mandarin may also have an enhanced response, which is thought to reflect, just greater utilization of pitch and tone.
Awesome. All right, and it does look like that's all the questions that we have.
Okay, so please join me in thanking Dr. Blair Kenshiro for a very interesting and engaging presentation. We are really glad that she shared her research and her team's research work with us. Thank you very much. For those of you who are listening this for PDH purposes, today's password is music. Okay, music. Thank you.